Hello guys, I am Ajay, a BSc Honors Agriculture student at Galgotias University. In this video, we are going to talk about rice blast disease. It is one of the most severe disease of rice. We will talk about what causes it, what are its favorable conditions, its symptoms, and how it can be managed. Talking about the causal organism of this disease, it is Pyricularia oryzae. The mycelium of this fungus is hyaline to olivaceous and septate. Conidias are pyriformed, hyaline, and usually three-celled. Talking about the symptoms of this disease. The symptoms appear on leaves, nodes, scratches, and glumes. On leaves, the symptom appear as boat-shaped or spindle-shaped spots, which have grey center and dark brown margin. And in favorable conditions, these lesions cover the whole leaf. On node, the node become brown, black, and dry. An infection at node often results in stem breaking. On neck, it is the most serious symptom of this disease. The neck of panicle turn brown to black and grains do not develop properly. The panicle become partially or completely sterile, or there is a formation of chaffy grains. These are its symptoms, as you can see on leaves, boat-shaped lesions, nodes turning brown to black, and on neck blast symptom, chaffy grains on the panicle. Talking about the disease cycle of this disease, the pathogen mainly survive on disease rice seeds, straw, and weed host. The hosts for this crop are Panicum repens or Echinochloa cruciale. Primary spread is through straw and weeds, and the secondary spread is mainly due to airborne conidia. Talking about the favorable conditions are intermittent drizzles, nighttime cooler temperature, high relative humidity 93 to 99%, and high nitrogen application. Talking about the management, first is cultural method, using of resistant varieties. Using resistant varieties against rice blast is the most economical way of controlling rice blast. Use of tolerant variety, avoid excess nitrogen fertilizer application, apply nitrogen in three split doses, remove weed host from the bunds because weed act as host for. Talking about the preventive methods, avoiding dry nursery, avoid late planting, avoid grasses or other weeds on bunds, seed treatment with Pseudomonas fluorescens at 10 gram per kg of seeds. Talking about chemical methods, at different stages, different steps are to be applied. At, at nursery stage, when there is light infection, spray carbendazole or edifenfos 1 gram per liter. Pre-tillering to mid-tillering stage of crop when their infection is 2 to 5%. Apply edifenfos or carbendazole at 1 gram per liter. Delay top dressing of nitrogen fertilizer when infection is seen. And panicle initiation to booting stage at 2 to 5% of leaf area damage, spray edifenfos or carbendazole at 1 gram per liter. And when the flowering stage appears or after the flowering stage when there is 5% leaf area damage or 1 to 2% of neck infection, spray edifenfos or carbendazole at 1 gram per liter of water. Thank you. That's all from my side. Have a nice day.